So, in this lecture we will be discussing the process of cell signaling and how it is relevant for the communication between two cells. So, overall this particular cell signaling process and cell communication is very important as far as the different cell fate processes are concerned. For example, cell migration, cell differentiation, cell division, cell proliferation. So, all these cell fate processes, one of the essential step is the cell signaling. So, if the cell signaling does not take place in a very appropriate manner, many of these cell fate processes cannot take place in under in vitro or in vivo conditions. In this perspective, that overall structure of this presentation will include, first I will go through some of the overview of the cell signaling processes, that will be followed by the various types of signaling. Then I will come to the different mechanisms of signaling. So, these mechanisms means like how these signaling processes takes place, how these signals are generated from the source cell and it is communicated to the target cell, what are the mechanism. Then fourth aspect is the quantifying various aspects of cell signaling. That means that the student should have an idea about that what is the time scale or what is the typical concentration over which a signal can be very effective and what is the concentration should be maintained. Then fifth one is that I will give you some examples particularly for the cell ECM interaction and another and different other cell signaling pathways. So, this is the overview of what I was just discussing 2 minutes ago, that is the tissue functions and underlying cell fate processes. Now, coming to the tissue dynamics like tissue function like homeostasis, like as I will discuss later that that all these cell survival, cell differentiation and cell motility, all these things are essential components of the homeostasis process. The second one is the tissue repair, that is the wound healing process. Third one is tissue formation like morphogenesis and developmental biology. Now, just few minutes back I mentioned that these are the cell fate processes, like this is the cell fate process like cell replication, that means cell division, cell differentiation like different gene expression or different cell functionality, cell death processes like necrosis or apoptosis, cell motion like cell motility and cell adhesion. So, all these processes cannot take place unless a given cell will receive particular and appropriate signals from another cell. So, that is what is mentioned, that cells communicate and coordinate their efforts to the cell fate processes. So, all these cell communication processes, the ultimate outcome is that cell fate processes. So, this is a typical eukaryotic cells like nucleated cells, and you can see that this process too means that these particular cells can be going to another state, that is the differentiated state. Then cell replication means these cells will increase in number. Cell adhesion means that it is an biomaterial substrate and on which there is protein adsorption as mentioned before, and this protein adsorption is important for the cell adhesion process. Cell motility means like a child walks on the ground, similarly how the cells will walk on the substrate or a given biomaterial substrate. So, like that cell motion, and how these cells like maybe I have mentioned earlier that cells have also filopodia or lamellopodia, like a human being has legs and hands. So, similarly the cells through lamellopodia extension or filopodia extension, the cells can walk on the material substrate. And number 3 process is the cell death, like cells will no more be surviving because of some problem either due to necrosis or apoptosis. Now, coordination of the cellular fate processes. So, cell communication essential to coordinate their activities and occurs, that takes place in 3 principal ways. Number 1 is that secretion of the soluble signals. So, here the soluble signals means that a source cell will generate or will essentially secrete some kind of soluble signaling proteins. So, signals means essentially they will generate some biomolecules or protein molecules or some other molecules, and these are known as signals. So, it can be cytokines or it can be chemokines. So, what is cytokines? Cytokines are actually growth factors. Now, growth factor, this term requires definition. So, growth factors are essentially cause proliferation and differentiation. So, these are like cytokines, and chemokines means growth factors that induce cell migration. So, essentially cyto means that is essentially related to proliferation and differentiation. Chemokines means which is essentially related to the cell migration. Now, again there are three types of soluble signals signaling processes. Number one is that autocrine. Autocrine means same cell signals to itself for the division, cell division processes. Like a mother cell is dividing into two daughter cells. So, therefore, a same cell can send the signal to the itself, so that cells can divide to make two daughter cells. Paracrine means cells signals to neighboring cells by diffusion, and endocrine means cells secrete some growth factor into bloodstream and carried into target cell. I will show these things, these three things in a few minutes. Then this is the soluble signals. The second one is that, so this is the number one. Number two is the secretion of the insoluble signals that alter the physical and chemical composition of microenvironments via modifications of ECM. So, insoluble signals. So, first of all, there is a difference between soluble signals and insoluble signals. Okay. And then second thing is that these cells they cause the changes to the physical and chemical composition of the microenvironments. That means, if there is a cell here, eukaryotic cells, this is nucleus and this is eukaryotic cells, microenvironments means this is called ECM, that is extracellular matrix. So, these insoluble signals cause some modifications to extracellular matrix. So, this is called another type of signals. Third one is that direct cell to cell contact. So, that takes place through gap junctions. Now, what is gap junctions? I will come to that in a few minutes. Okay. So, this is direct cell to cell contact, and fourth one is the response to mechanical stimuli in their microenvironment, equivalent to biochemical stimuli. So, the first three are three different principal ways of the cell communication. I repeat, that is through soluble signals, second one is the insoluble signals, third one is the direct cell to cell contact. Now, as I said that what is growth factors? So, growth factors are essentially small proteins that are on the order of 15 to 20 kilodalton. So, Kd stands for kilodalton, that means 15 to 20,000 dalton. Now, what is dalton? 1 dalton is equivalent to the weight of one hydrogen atom. Okay. So, therefore, growth factors are nothing but the small protein molecules which has a molecular weight of 15 to 20,000 kilodalton. Okay. So, this is the growth factors. Now, there are different type of growth factors. Since it is not a course on biology as such, I would like you to remember that at least 2 or 3 different growth factors which are important in the process of cell fate processes. Number one is that fibroblast growth factor, that is FGF. Number two is that insulin like growth factors, that is called IGF, and third one is that transforming growth factor, that is known as TGF. Now, what is the functions or biological activity of these 3 growth factors? Now, fibroblast growth factors, they are essential biological activities mesodermal and neuroectodermal cell stimulator family of about 19 similar proteins that play a role in skeletal and nervous system development. So, these particular growth factor, that is the fibroblast growth factor, they essentially help in the development of skeletal and nervous system nervous systems. Insulin like growth factor IGF, that stimulates proliferation and differentiation of different growth factors. So, therefore, as you can see in the context of the cell fate processes, IGF is more important because IGF essentially stimulates, or it facilitates cell proliferation and cell differentiation. Transforming growth factor beta, that regulates cell growth and differentiation of many cell types involved in regulating extracellular matrix proteins. So, TGF actually regulates cell growth and cell differentiation of many cell types. So, as I said last slide that 3 types of growth factors are important. I repeat, FGF is fibroblast growth factor, IGF is that insulin growth factor, and TGF is transforming growth factor. And out of that, IGF is the main growth factor which is important for the cell proliferation differentiation. Now, 2 or 3 slides back I mentioned that what are the different types of cell signaling process, like one is that contact dependent, that is that through gap junctions, and then 3 other types of signalings that I have mentioned that is through soluble signals. So, if you remember correctly, there are three ways this signaling takes place. One is that soluble signals, second one is that insoluble signals, third one is direct cell to cell contact. Now, under the category of soluble signals, there are three different ways this signaling can take place. One is that autocrine, one is called paracrine, and third one is called endocrine. Autocrine means cells signals to itself. Paracrine means cells that signal and then it will go to another target cell and then it is the paracrine. And endocrine means cells secrete the signal molecules that will be carried away through the blood stream and then it will be transported to another target cell. So, that is the paracrine. Now, here you can see that is the first one is the contact dependent. So, this is your signaling cell S and this is your target cell T. So, from signaling cell the signal molecules you can see membrane bound signal molecule, that is the red one, and this is your target cell, green one is your target cell surface receptor. So, they will come and then they will attach, they will get attached to the cell surface receptor on the target cell and then they will cause some changes in the target cell T. So, this is like contact dependent, like two cells are in direct contact, and among various cell types, the cell type that comes to your mind where this contact dependent signaling takes place is that endothelial cells. Endothelial cells like cells which are arranged one after another in a very dense manner. Paracrine, this is your signaling cell S. Okay. Now, there are 2, 3 or different target cell. This is 2, this is one target cell, this is another target cell, this is T1, this is T2 and this is T3. Okay. Now, these are all eukaryotic cells as you can see. Now, this signaling cell they produce or they generate 2 or 3 different type of signaling proteins for example. Now, one of the signaling proteins can be transported to this T3, but via local mediator. That means, it cannot directly be transported via protein molecule directly to another targeting cell. So, this red one will get attached to the local mediator and then this local mediator. So, this local mediator can be transported to the target T3 cell. Similarly, different local mediators are present, as you can see, this is another local mediator and this is another local mediator. So, the signaling cell from S to the target cell T1, T2, T3, the transport of the signal molecule takes place via the local mediator. So, this is the process of the paracrine signals. And endocrine signals means this is your endocrine cell S, that is the source cell. Now, this is your target cell, this is T1 target cell, this is T2 target cell, all are eukaryotic in nature. Now, endocrine cell this secretes some signal, then it will go to the hormone and then through the blood stream. This is your blood vessels or blood stream, it is going on and then it is transported to the blood stream and then it goes to 2 target cell. So, this is like T2 and T3. So, essentially all you can see here that from S the target cell goes to T1 and T2 and then it can be transported. So, this is called endocrine signals. Okay. And then another one that has been mentioned here is that synaptic signal. Synaptic signaling is performed by neurons. So, neurons means it is actually important in neural tissue engineering or in general neurons. So, this is the transmit signals electrically along their axons and release neurotransmitters at synapses. Okay. So, here you can see these are like synapses here. Okay. And these synapses there is a neurotransmitter. So, these signals as such it is transported electrically and then it is going to another target cell. This is your source cell and this is your target cell T. So, from S to T that through neurotransmitter this signals is transported. Okay. So, direct cell to cell contact, this is little bit more details on the direct cell to cell contact. Like eukaryotic cells, they express proteins on their surface that lead to direct cell to cell contact, enabling highly specific communication. Highly specific communication means if you go back here, that signal molecules that is generated here from this S cell, these same signal molecules can be transported either T1, T2 or T3. So, it can be in nature non-specific, but when these two cells are in direct contact, you know that these signal molecules which are generated from this S cell, it will directly go to the T cell, because just because it is physically in contact with each other. So, this is called highly specific communication. Highly specific communication means this signaling communication is exactly for this T cell, target cell, not that another target cell which are located here. So, then in contrast, soluble and ECM signaling is non-specific. Soluble and ECM signaling means, as you can see, this is called soluble signaling, or this is another soluble signaling, that is paracrine and endocrine. So, these signals, the source cell does not know where their signaling proteins will finally arrive at, whether it will arrive at T2 or whether it will arrive at T1. So, that means this is like non-specific. So, I hope you are getting my point, that these are specific means just because they are contact dependent, direct. So, source cell to target cell, this is called specific interaction. Non-specific interaction means like source cell, it is going to two target cells, and this is called non-specific interaction. So, it is non-specific since the signaling cell has no direct knowledge of the target cell and vice versa. Has no direct knowledge means signaling cell does not know which target cell their signal protein will finally arrive at. Okay. Then third point that has been mentioned here is that cell to cell signaling molecules come in two principal forms, like those that allow mechanical contact between two cells, and then secondly those that form junctions between cells through which molecules can be passed directly from one cell to the next cell. So, this is what I was mentioning there, that signal. So, this is signaling via gap junction. So, this is the direct contact, and this direct contact here you can see these are the signal molecules. Okay. So, these signal molecules, it can go from source to target, and this is highly specific in nature. Highly specific in nature, why? Because this, if this is source and this is target, source cell exactly knows this is the target cell where the signaling molecules will directly pass through, and these junctions here it is known as the gap junctions. Now, these gap junctions, the communication through gap junctions only take place in cell type like endothelial cells, where multiple cells are lined up one after other. So, there is a direct physical contact between the two type of cells. So, here it has been mentioned that 2 points. Point number 1 is that cells connected by gap junctions share small molecules and can respond to the extracellular signal in a coordinated manner. So, can respond to the extracellular signal in a very coordinated manner. And second point is that some of the molecules involved in direct cell to cell contact, known as cell junction molecules, allow for direct cytoplasmic communications. So, like molecules who are in direct cell to cell contact. So, this is your cytoplasm C, this is your cytoplasm C here. So, these signal molecules, if it is transported between these or in a reversible manner, can directly communicate to the cytoplasm. Now, coming to the some quantification, estimating that intracellular fluxes. Now, how to quantify that, what is the typical fluxes that will be generated in this intracellular in this signaling process. So, let me go back. So, intracellular fluxes means like we need to quantify that how much signal molecules that will be generated at the source cell surface here, and how much flux that will be generated, and that will be going to the 3 different cells which are mentioned here. So, that means what I am trying to tell you is that what would be the flux of the target, what will be the flux of the signal molecules that will be transported from one signal cell to multiple target cells. So, now for the particular case of the cell to cell junctions, typically cell to cell junctions, their dimension is around 1.5 nanometer, and therefore, that allow below 1000 Daltons. So, most of the growth factors, if you remember, they are very small protein molecules, and which has a size of 15 to 20 kilodalton, means like 15 to 20,000 Dalton. So, therefore, all these growth factors can be easily transported between these two cells, and these 1000 Daltons to pass between cells. Now, kinetic theory shows that the flux through a hole of diameter d of a solute with a diffusion coefficient capital D that is present in the signaling cell at a concentration C1 and the receiving cell has a concentration C2. So, this flux is J is equal to 4 D, D is the diffusion coefficient here, small d is the cell to cell junction diameter, C1 is the concentration in the source cell, C2 is the concentration in the target cell. Receiving cell means this is essentially target cell, and signaling cell means this is essentially source cell. You understand what I am saying? Let me go back to this. Okay. So, what has been mentioned here, suppose this is source cell and this is your target cell. Here the concentration is C1, here the concentration is C2. Okay. Capital D is your diffusion coefficient, that how this signaling cell will be passing through, and this diameter if it is small d here. So, then the flux, that how much flux that will be transported, that has been calculated via simple fixed law, is J is equal to 4 d pi over d C1 minus C2. So, always the flux will be from higher concentration to the lower concentration. So, now if you see that if you consider that d is 4 nanometer, capital D is 10 to the power 5 minus 5 centimeter square per second, like typical diffusion coefficient, C1 minus C2 is 100 micromolar. Then what would be the typical flux? This typical flux would be 2.4 into 10 to the power 5 molecules per pore per second. So, let me go back to this slide and let me explain to that. So, this small d has been assumed as, suppose this is 4 nanometer. So, if you calculate here d is equal to 4 nanometer, your capital D diffusion coefficient is 10 to the power minus 5 centimeter square per second, and your C1 minus C2 is typically like 100 micromolar. Okay. Then this flux, which is F, it is like 2.4 into 10 to the power 5. So, flux is around 2.4 into 10 to the power 5 molecules per pore per second. So, let us like understand these numbers. Okay. So, these numbers means, that means if there is, this is the particular one gap junction. Okay. So, through these gap junctions from one cell to another cell, around 10 to the power 5 orders of molecules will be transported each second. Okay. So, that means if you now consider that cell culture time frame is typically few hours, like 24 hours to 72 hours, and typically when cells will adhere initial stages, that takes place maybe for 6 hours. So, for 1 hour means 3600 second. Okay. So, in 1 hour how much molecules that will be transported? 2.4 10 to the power 5 multiplied by 3600 multiplied by n, that capital N is the number of pores that are present among the two different cells. So, you can see that the amount of molecules that will be transported, which will be huge number, it is like around 10 to the power 8 molecules per pore in 1 hour, and 10 to the 8 molecules per pore means depending on the cell type, that number of pores you can consider. So, it can be 10 to the power 9 to 10 to the power 10 order of molecules that will be transported per hour. So, that is a huge number of cells. If they will be transported, those huge number of molecules, those will be transported between two cells. Now, you can see typically in culture medium, you culture at least 10 to the power 4 to 10 to the power 5 number of cells. So, huge number of cells are cultured on a material substrate. So, now you can understand this entire process, that your total number of cells is 10 to the 4 to 10 to the 5, your molecules that will be transported between two cells, which is of the order of 10 to the power 8 to 10 to the power 10. So, such a large number of molecules are involved in your cell adhesion process, cell proliferation process, cell differentiation process. Okay. So, this you as students, one should have a feel for the numbers that will be responsible for certain biological processes. It is not the exact number like 2.4, but it is the order of magnitude like 10 to the power 5 or 10 to the power 8 or 10 to the power 10 molecules that are responsible, or that is this molecular transport is responsible for certain processes. Okay. So, this is what in the initial slide I mentioned that one should quantify the total number of one should quantify the intracellular flux, that how much flux is there and how these fluxes can be calculated and so. Now, here it has been mentioned, next point is that with approximately 100 pores between cells, between the two cells, if you consider 100 pores, flux will be of the order of 2.4 in 10 to the power 7. So, that means 2.4 10 to the power per second per cell boundary per second, that would be the molecules that will be involved in this cell signaling process. And molecules passing through two cells are restricted to 1 kilodalton, that I have mentioned here, that is 1000 daltons. So, extremely small molecules, they can be only transported by direct cell to cell contact. If the molecules are larger in size, then what would be the alternative way? Then it can go to paracrine or endocrine, like you remember this paracrine and endocrine, that means this is the paracrine, like there will be soluble signals, they will be soluble in ECM, or endocrine means they will be soluble in bloodstream and they will be transported to 2 or 3 different target cells. Okay. This mechanism is extremely important in passage of electrical current for cells, for example, cardiomyocytes or cardiac myocytes. So, cardiomyocytes are the cells which are typically cultured on the heart tissue replacement, or if you want to develop some cardiovascular patches, for example, PLGA carbon nanofiber composites and other similar composites. If one has to evaluate that whether these materials are suitable for the patches to be developed for the hearts. So, there in order to find out the cytocompatibility, people normally culture the cardiomyocyte cells. The other things that I mentioned in the overview slide is that there are 3 types of signalings. One is the soluble signal, one is that insoluble signal, third one is the direct cell to cell contact, fourth one is the mechanical stimulus. Now, mechanical stimulus means the cells and tissues respond to mechanical stimulus like shear stress or compression or tension. Now, people from material science background, they always think that shear stress, compression and tension should be of the order of megapascal or gigapascal. Okay. But one has to understand that cells are extremely compliant, they are not very stiff, and typically the cell's elastic modulus is very low, if the order of kilopascal, like 10 to the power 3 Newton per meter square. So, therefore, the shear stress and compression and tension, these stresses also should be of similar order, like kilopascal or small or extremely small force, like. So, here you have to understand, if you are considering 10 to the power 3 Newton per meter square, therefore, that your force that one should apply to understand the behavior of the cells under force, under compression or tension, this force should be of the order of piconewton or extremely small load. Okay. Otherwise, you cannot study these cells. Then integrins act as mechanical links between the extracellular matrix and cellular cytoskeleton and likely to play a major role as force transmitters. Third point is that exact molecular mechanism that transduce mechanical stimuli to biochemical events have not been elucidated yet. Fourth point is that it is clear though that shear stress will change endothelial gene expression, that compressive forces will alter chondrocyte matrix metabolism. Now, endothelial cells are the tissues or the cells of the endothelial tissues which actually prevent the different body parts, and chondrocyte cells is the cells which are useful for cartilage. Now, when you have this cartilage fracture or cartilage breakage, so there you need to seed the chondrocyte cells on the cartilage matrix, so that the cartilage can grow. So, here it has been mentioned that shear stress will change endothelial gene expression and compressive forces will alter chondrocyte matrix metabolism. So, essentially these examples essentially illustrate that how mechanical stimulus, like in terms of the tensile, shear or compression stresses, they can locally change the cell fate processes like gene expression or metabolism, etc. Now, autocrine signaling means that cells they will signal to itself. So, here you can see this is your source cell, a single signaling cell receives a weak autocrine signal, and then this is in a group of identical signal cells, each cell receives a strong autocrine signals. So, here suppose this is your source cell, but all these cells they are like similar like to source cells. For example, you are culturing only fibroblast cells. So, all the cells are of similar nature, fibroblast only. Okay. So, these are like L929 cells. So, therefore, here in case of the autocrine signals, or a single cell which is at the center of around 6 cells here, they will receive similar type of signals which will be generated by all these 3 different types of all these 6 cells which are of similar nature, which are of also fibroblast nature. So, essentially that what is the physical implications of that? A group of identical cells, they will produce a higher concentration of a secreted signal than does a single signal, and when the signal binds back to a receptor of the same cell type, it encourages cells to respond coordinately as a group. Now, when this autocrine signaling happens, like you take an example that suppose you do not know that whether material is cytocompatible. So, then you are seeding all fibroblast cells like 10 to the 4, 10 to the 5 fibroblast cells you are culturing or you are seeding on the material. So, that means there you can expect autocrine signaling. Why? Because individual L929 cell will receive signals from a few other neighboring L929 cells, and therefore, the strength of the signals or the concentration of the signal molecules also will be higher, and that is what I am trying to convey to you. So, therefore, in a group of cell, a single cell, let us take an example which is star here. This single cell will receive the signal molecules from all the similar cell type which are in its neighborhood, and the concentration or the strength of the signal also would be higher. Now, sending a paracrine signal. So, this is your signaling cell or source cell, this is your target cell. Okay. Now, signaling cell, source or target cell, at the signaling cell surface your signal strength or concentration. So, this is also in biological language it is called signal strength. Okay. So, this signal strength is the highest, but then it will go to this target cell via molecular diffusion process, right. Molecular diffusion process means the signaling cell that it is generated. So, these are like all soluble signals, okay, because it is not autocrine, it is paracrine signals. So, these are like soluble signals. Now, once the soluble signals will be transported from source cell to target cell, then what will happen? That with distance this concentration also will decrease. So, at the target cell your concentration is this one, okay, and your source cell your concentration is this one. So, signal strength also will decrease because your signal concentration or concentration of your signal molecules decreases. So, therefore, a target cell senses signal depending on its location relative to signaling cell and its sensitivity to the signaling molecule. Sensitivity means that how well this target cell is equipped in terms of the cell surface receptor. So, these are like cell surface receptors in terms of the cell surface receptors, so that the signal molecules once it is transported from source cell to signal cells, they will be able to bind to the receptor of the target cell. Okay. So, that means that is known as the sensitivity in biological language. Once sufficient number of signaling molecules binds to receptors on surface of target cell, they induce a signal transduction process. Now, what is signal transduction process? That I will explain to you after few minutes. So, from this point it is very clear to you that source cell or signaling cell, your concentration of the signal molecules will be at the highest level. Once it will go through the molecular diffusion process to the target cell, then this concentration will decrease. However, these whatever molecules that will be transported by molecular diffusion to target cell, the target cell also should have a good affinity towards binding to this molecules, signal molecules. Okay. So, that has been mentioned by the sensitivity to the signaling molecules Km. Now, sensing a concentration gradient, now given a cell size typically 10 to the power 10 to 25 micron and no concentration of signal molecules, sometimes greater than 1000 molecules, it is difficult for a cell to sense a concentration difference across a cell body. So, what it means here, typically the cell size, this cell size is around 10 to 25 micron. Okay. So, that is the diameter, okay, of the cells. Now, if the signal molecules here, suppose the concentration that is reaching here 1000 molecules, that means whatever the molecules that are generated at the source cell, only 1000 molecules are coming here in the target cell. Sorry, now these 1000 molecules which are coming here in 10 to 25 micron size cell. So, what has been mentioned here, that it is difficult for a cell to sense a concentration difference across the cell body. Means, what it means, that inside the cells also there are a lot of molecules are present, and suddenly this cell has to sense that 1000 molecules which are coming by transporting from the molecular diffusion from source cell. It is difficult for a cell to sense these 1000 molecules. So, what it does, it says that a cell can extend podia to travel the distance over which it is determining a concentration gradient. What it means? This means that this target cell T, it will extend its filopodia or lamellopodia. It is like stretching a human being's hands or legs. So, once this lamellopodia or filopodia, there is a leg kind of things extension is there. So, this lamellopodia can sense more higher number of concentration here, because the concentration gradient is down this way. So, are you able to get it what I am saying? What I am saying, the cell itself will not be able to sense, but cell will extend its filopodia to some distance, so that this filopodia can sense a larger number of signal molecule concentration which is present. In continuation with the discussion that I had in earlier lecture, so let me repeat what I said in the last lecture, that that there is two cells, one is the signaling cell, one is target cell. Now, if the target cell is located far from the signaling cell, so that the concentration of the signal molecules that will be sensed by the receptors of the target cell is very small, for example 1000 molecules or so, then what will happen? This target cell will extend its legs or hands just like a filopodia or lamellopodia. Now, once this filopodia will be extended towards the signaling cell, then it can sense a higher molecule concentration or higher concentration of the molecules, so that there will be more effective binding of the signal molecules to the cell surface receptors. Okay. So, this is what exactly happens in case of the target cell which is located far from the signaling cell. Now, filopodia or neurites are highly dynamic structures that are used to guide the migration and growth of a cell in a concentration field. Now, what are the maximal secretion rates of the proteins? The maximal secretion rate is around 2000 to 8000 antibody molecules per cell per second. Now, this corresponds to typically 1 picogram per cell per hour. 1 picogram means 10 to the power minus 12 gram. Okay. Nanogram means 10 to the minus 9 gram, picogram means 10 to the minus 12 gram. So, one individual cell, what it means, one individual cell can secrete 1 picogram of the signal molecules per hour. So, if you go back to the slide, this particular slide, so particularly this source cell, so this can secrete 1 picogram of signal molecules in 1 hour. So, if you culture 24 hours, so how much picogram of these signal molecules that will be generated by individual cell? It is 24 picogram. Now, if you culture 10 to the power 5 cells, then what would be the total number of signal molecules that will be generated? Typically 24 multiplied by 10 to the power 5 picogram. 10 to the 5 into 10 to the minus 12 is 10 to the minus 7. So, that means it will be 2.4 into 10 to the power minus 6. So, total signal molecules that will be generated will be 2.4 into 10 to the power minus 6 gram, right, in 1 hour. So, 2.4 into 10 to the minus 6 means 2.4 microgram. So, from picogram to microgram, that is like a few micrograms of the signal molecules that will be generated. Remember, these signal molecules are extremely smaller in molecular weight, like 1000 kilodalton, 1000 Dalton or so. So, these small molecular weight signal molecules that will be secreted by the source cell. The next question that I have posed here, that how far can soluble signals propagate? That means, what would be the typical distance d, or what is the typical distance s, for example, d or s, over which these signal molecules can be propagated. Now, here there has been very simple calculation that has been done based on the molecular diffusion theory. So, under steady state conditions, concentration C of a secreted molecule as a function of the distance r from a cell. So, function of a distance r from a cell means this is your Kd. So, Kd is the dissociation constant of a signal molecule to receptors on the receiving cell. C is the concentration of the molecule. Small a is that another one is the tau diffusion by tau secretion, and capital R is your cell size and small r is your distance. So, what you see here, the concentration is inversely proportional to the r, that is the distance from the cell. Okay. So, what it means? Higher the distance, lower will be the concentration. Is it clear? So, what I am saying here, this concentration is inversely proportional to small r. Small r is the distance from the cell. So, here in this particular diagram, if you look at here, so this small r is this. So, this is your small r distance, okay, that is the distance from the cell, and this is your concentration C here at the source cell. So, if C proportional to r means the way it will go, the farther you will go, you will sense lower concentration of the soluble signals. Are you getting my point? So, therefore, that larger the distance, smaller is the concentration, and A is that R critical by smaller. So, maximal secretion rate is given by the ratio of two time constants. One is the time constant for diffusion, this is the tau, and for producing signals, and another one is the secretion time constant, that is the tau secretion, that is KDRV. Now, concentration at signaling cell surface, or that is the source cell surface, that concentration is the C surface is equal to RFD by this, and then you can simply do some arithmetic. And what is the physical meaning of all this expression? Physical meaning is that the highest concentration of the signal molecule is found at the signal cell surface, and that it decays in an inversely proportional manner with distance from the signaling cell. So, this is the physical implication that you have to bear in mind. That means, the signal molecule concentration will decrease as you go away from the one source cell towards another target cell. A reasonable estimate of the signal propagation distance is the characteristic distance at which the signal strength is half of its maximum, leading to a time constant estimate of about 20 minutes and a maximal distance of about 200 micron. So, what it means, let me just explain to you here. So, this is your source cell, this is your target cell. Okay. So, this particular expression, what it says, suppose your source cell here the concentration is C naught, signal molecule concentration. Now, your target cell minimum concentration should be C naught by 2 for the signal molecules to be effective for this target cell. Okay. And if you do some calculations, considering that typically it is 10 to 25 micron is the cell diameter and so on and so forth, then what you find that the critical distance through which these cells will be able to communicate to another cells is typically 200 micron. Okay. So, therefore, when you seed the cells in a typical culture plate, when you seed 10 to the power 4, 10 to the 5 number of cells, instead of seeding 10 to the 4, 10 to the 5 number of cells, you just seed 10 cells. What will happen? It will happen like cell to cell distance would be much higher than 200 micron. So, 10 cells they will not be able to communicate to each other just because their distance between two neighboring cells is too far. So, there will be absolutely no process of cell division, cell growth and cell proliferation. It is clear? So, this distance is altered proportionately to the secretion rate and inversely with the dissociation constant. So, this critical distance whatever you can say, that is R critical, which is around 200 micron. So, this R critical is typically proportional to the secretion rate F. So, secretion rate F means the more the source cell will secrete large number of molecules, then it is possible that even if the target cell is farther from the source cell by more than 200 micron, let us say 500 micron or so, then also target cell will receive a critical concentration of C naught by 2 number of molecules at the target cell. Okay. Let me repeat what I said. What I said is that suppose your secretion rate is too high, secretion rate at the source cell, this is your source cell, here the secretion rate increases, that means the S cell will secrete more number of molecules, signal molecules, right. Now, I said that in the normal circumstances the target cell should be preferably at a distance of 200 micron or less. Now, your F will increase, that means your signal cell secretion would increase, then what will happen? Then even if the target cell is here, let us say around 500 micron, then also in the target cell your signal concentration may be C naught by 2, so that that signal may be effective for the target cell, understand. So, the critical distance would increase in that case if the secretion rate will increase. So, the another factor is that internalization of the growth factor by target cell. In many cases the receptor ligand complex, receptor means cell surface receptor, ligand means that is the signal molecule, is internalized, and that means signaling molecule dissociates from receptor and receptor recycled on the cell membrane. A typical time constant for internalization of signaling molecules is of the order of 15 to 30 minutes. Now, why this time constant is important and these numbers are important, that means, so here is your cell surface receptor, this is your signal molecules. So, what I am trying to say here, signal molecules will come, they will interact with the cell surface receptors here, okay. And then it may so happen that once it binds, it can get dissociated from the cell surface receptor and it can be internalized also in this cell. Now, typical time constant means the time scale over which this association and dissociation that take place, that is of the order of 15 to 30 minutes. Now, this time scale is important because this time scale, from this time scale you can understand that why your typical culture time is not 2 hours, but it is 24 hours or 72 hours. That means individual cells, if you want to activate by signaling, for individual cells it will take 15 to 30 minutes. Okay. Now, there are let us say 10 to the 4, 10 to the 5 number of cells and it takes place in a very random manner. So, you can say that large number of time you required, several hours before all the cells will be activated. So, that is the reason you may find that after you culture on a same substrate, it is not that all the cells they have proliferated or they have expanded in the similar manner. Some cells they have expanded much significantly, some cells they have started expanding. Why there is a response difference? The response differences means that individual cell which has expanded very significantly, they might have received large number of signals from the neighboring cells, whereas the other cell which did not expand significantly, which did not proliferate significantly, that means simply because that cell might not have received large number of signals like the other cells. So, because of the difference in the signaling processes or because of the difference in the amount of signal molecules that they have received, the cells also cannot proliferate or cannot grow. So, therefore, all the cell fate processes of the 10 to the power 5 cells that you are culturing on a substrate, they are different just because that individual cells they have received different amount of signal molecules for their activation for the cell fate processes. Okay. So, the absolute value of the growth factor uptake rates have been measured, and this is for IL-3 and SCF. Now, this is like insulin growth factor, you remember. So, growth factor means, so this growth factor, definition of growth factor is 15 to 20 kilodalton. So, small protein molecules whose sizes are 15 to 20,000 kilodalton, 1 dalton means atomic mass of one hydrogen atom. Okay. So, they are consumed by intermediate hematopoietic cells at a rates of 10 to 100 nanogram per million cells per day, and 10,000 to 70,000 growth factor molecules need to be internalized to stimulate the cell division. That means, you can see now that 15 to 20 kilodalton that whose growth factors are there, and this is like 10,000 to 70,000 growth factor molecules need to be internalized. Internalized means need to be taken inside the cell in order to promote the cell division, like cytokinesis, mitosis, etc.